Thank you for listening. I will be presenting some of our results from our grant entitled Towards Sustainable Management and Cultivation of Macroceases Pirifera in Peru, supported by GlobalSeaweedSTAR. And this project was managed by the Universidad Peruana Cayetano Heredia in Peru, with the participation of the Universidad Católica del Norte and the Universidad de los Lagos, and also that includes several institutes, allowing us to have a large group of researchers participating. In this proposal, I wish to start just by mentioning that we share in the north-central part of Chile and Peru almost the same kelps, mainly different type of species of Lawsonia, and Macroceases pirifera. Why our interest? Well, mostly because this kelp show a high plasticity, high morphological variability, reproductive phenology, different populations, variations in the population's dynamic, and different relevant biological interactions that explain their abundance. Also, we have found that there is a genetic variability along latitudinal gradients, but we don't know what is going on in Peru. We know that we have a differential responses of photosynthesis, nutrient uptake, growth of macroceases between locations and between, so local adaptation should be relevant, but we are willing to see if we see some patterns along the whole latitudinal range of the southeast coast of Chile, Peru. The main objective was to have first a training course for Peruvian young researchers there, and also a general open workshop. So the access to information to undergrads and graduate students in Peru and Latin America could be also being reached. The research about population genetic, epiphyte pathogens, potential pathogens, and some description of the some morphological and abundant traits of macroceases along the Chilean and Peruvian region was also our main targets from this perspective. We started in October. There is something wrong, 2020, with the training course mostly for the people that was engaged to do the sampling in the field. So we discussed about sampling protocols, the kelp population dynamics, genetic diversity, epiphytes, and how we need to deal with our sampling protocols to get good data on these subjects. So we had another workshop on December 2020 about a global ecology kelp perspective, and in which we discussed about ecology, economical importance of kelp, then some ecological population dynamic of Peruvian and Chilean macroceases populations, what we know about genetic and kelp evolution, the importance of some abiotic and biotic factors that determines kelp abundance, and we lastly discussed and presented information about kelp farming in the southeast coast of South America. So we started with the research in the north of Peru, and we moved south until Ilo, which is near the Chilean border. In these 6 locations, we sampled Macrocystis sporophytes, 30 individuals, and each one was divided in different portions, basal, apical, intermediate, to sample for epibionts, potential pathogens, and some morphological characteristics of the algae. So we measure morphological and reproductive traits. We took tissues for genetic analysis, and we did a photographic sampling of epibionts and pathogen symptoms on the sporophytes of the different populations, so 6 different populations. Just to have a snapshot on our results, we took different type of measurement about abundance, size, some morphological traits of the kelp populations in these different locations in Peru. And we did not find any clear pattern along the latitudinal range. We expected that further north, individuals could be could show a lower or smaller sizes, but this was not a clear case with our results. There was a lot of variation, and we did not reach to any conclusions. About the biomass, we have here the average and the standard error of the biomass that we found along this coastal gradient. And again, the pattern, we do not have a north-south pattern. There is local variation mostly. There's places with a lower abundances. Marcona had the highest abundances and some intermediate values, but also we could find in the extreme some significant variations, but between all the other sites, there is no variations, indicating that local conditions are more relevant than the variations along the latitudinal gradient. We took some photographic samples to determine the epibionts that we identified by taking samples in the field. And with these, we could find that the presence of epibionts on the sporophytes that were collected in these 6 locations were most prevalent in the north than in the south, where we had the lowest amounts of epibionts. About the potential pathogens, we have here symptoms that we can observe on the sporophytes that we collected. And in this sense, we can indicate that in general, we do not have a tendency that some populations, north or south, have more or less prevalence of symptoms that may be associated with some potential pathogens of giant kelp in the southeast coast of South America. And these variations varied from lowest values of about 50% of prevalence in the sporophytes and almost up to 80% of the sporophytes in some other locations. So thank you to Global Seaweed Support for allowing us to do this work. Cooperations between Peru and Chile is not always easy to find ways how to support this work, and this opportunity was very relevant for every one of us that participated in this research and in these training courses. Thank you very much. Bye-bye.